Good evening. I'm Dr. Richard Garassi. I think most of you know, but some of the community folks may not. President of Wagner College, and happy that you're all here in a large turnout. You're going to hear an incredibly interesting talk tonight. I had the privilege of meeting Dr. Monk a number of months ago when she gave a talk on behalf of raising funds for neuroscience research at Columbia University Medical School at a club in New York. And I was so taken by her talk for a number of reasons. One, you'll find it incredibly interesting, and she uses a lot of multimedia and the like to keep us engrossed. And I'll let her talk about her talk. I won't give you the spoiler. But what I also found was that she mixes the disciplines. She's so creative in the way she uses a lot of interdisciplinary stuff as well as stuff in her own field. And as you know, she studies development of the brain in the fetus, which is really, really fascinating. And I was also impressed with her, and particularly I'm happy that so many students are here, because I found her just a remarkable role model of a young professional, really dedicated to her research, but also, and this is the bonus for me, was that she does theory and practice. She uses her research to actually develop a practice working with families and children, in addition to doing the major research, primary research that she's doing on the brain. So I thought that was perfect for Wagner, because everything we're about in the Wagner plan is theory and practice, applied learning connected to a theoretical context. So I'm going to get out of the way and introduce Dr. McNair, who's going to introduce Dr. Monk. So you all know Dr. McNair, our wonderful provost, who is a wonderful transformative leader in her own right. I'm so pleased that she's with us. So here's Dr. McNair, a clinical psychologist.
Thank you very much, President Garassi. You are all in for a treat. As we had dinner with Dr. Monk, and students and faculty alike remarked on the exceptional work that you're doing, the interdisciplinary nature of your work, and as you listen to her describe her research, especially for those of you who are students, think about what we mean when we talk about the importance of working collaboratively with others from different disciplines and perspectives to tackle a unique problem. You are going to see that living in action as you hear her talk. Dr. Catherine Monk is professor of medical psychology in the Department of Psychiatry and OBGYN at the Women's Program at Columbia University Medical Center. She is also Research Scientist 6 at the New York State Psychiatric Institute. She was originally trained as a clinical psychologist treating children and adults in a program that emphasized the developmental origins of psychopathology. She then completed her postdoctoral research training in the psychobiological sciences via a National Institutes of Health Fellowship at Columbia University in 2000, and shortly thereafter began joining the faculty there. She is a 2-time recipient of the very prestigious Brain and Behavior Research Foundation Young Investigator Award, and her research has been continuously funded by the National Institutes of Mental Health since she got her first support as an NIH-K Career Development Awardee in 2001. She brings together research from the fields of perinatal psychiatry, developmental psychobiology, and neuroscience to focus on the earliest influences on children's developmental trajectories, those that happen in utero, and how to intervene early to prevent risk from mental health disorders in the future. I worked very hard not to ask her what those interventions are. We're waiting for the extraordinary kicker here. She is internationally recognized for her contributions to the developmental origins of health and disease research model, and most recently, she's been awarded key roles on the NIH-wide ECHO project, which stands for Environmental Influences on Children's Health Outcomes, a 7-year national-wide effort to study early factors, including prenatal and children's health outcomes across 50,000 participants. She's a primary investigator on one ECHO award, investigator on another, and elected by her peers to a 2-year term on the ECHO Executive Committee. We have here an outstanding and accomplished researcher who is going to speak to us about pregnant women's mental health matters for her and her baby. Please join me in giving Dr. Monk a warm Wagner welcome.
Really such an honor to be here, and thank you for such warm introductions. And now we just have to do a little furniture rearranging here so I can see everything. And I have to say I'm old enough now that I haven't had the word young next to my name in a while, so if anything happens tonight, that's what I'm going to hold on to. It's the best moment. It's really thrilling. And I do hope, speaking of being young, that it's been such a pleasure to be at this school, and the warmth and energy of this environment is terrific. I did have the pleasure of talking to several students. I would really welcome anyone, particularly students, asking me, how I got to be where I am and what were the bumps in the road or anything like that. I'd really be a privilege to talk about it. Okay, so you know the topic, and we had a fun time this afternoon seeing if everything's going to work. So we'll see if this works, and if I push this button, it worked. Okay, so there are going to be three major foci of my talk tonight. And I'm really interested just to emphasize in questions, but I'd prefer to take them at the end. So we're first going to be addressing the topic of a hidden yet receptive fetus. And the possibility already has been indicated that women's experiences, particularly their mental health experiences, may be transmitted to the fetus. And how does that happen? And because this does happen, I'm going to try to convince you with some evidence, some data that it does. What can we do about it? That this is a prime opportunity for intervention for both the mother and her future child. So, before we are as we are now, before this, and this, and this, and this, there is pregnancy. Pregnancy is the first period of the mother-infant relationship. It's also the first period of gene-environment interaction. Both contribute to future child development. That's right. We cannot easily get access to the fetus. But the fetus is experiencing the mother's life. And I want to give you a sense of that. This is a fetus. Maybe some of you in the PA program or nursing have seen this. This is a fetus, latent pregnancy. That's the heart rate. It's rather fast, 120 to 160 beats on average. And what you're seeing there is cross-section of the arm moving. The head is on your left. The feet are coming out on the left. It's a wonderful sound. Now it's going to be quiet. And you're going to see the cursor is right there on the fetal heart rate. You can really see that. And you're going to get a glimpse of the skull. And you can see the profile for a minute there. And you should be really seeing that heart beating now. There's a good picture. So this is a hidden yet receptive fetus. The fetus's brain development. Oops, is that getting cut off for you too? Oh, that's too bad. We had to switch computers. The fetus's brain development means that they can perceive aspects of the mother's life, and they can be affected by what's going on in the mother's experience. Now, is there something playing in the background? Does other people hear that? I'm sorry? Oh, it's the other classroom. I just have very good ears. Okay. So we need to think about fetal brain development right now, given that the fetus, again, can register what's going on in the mother's life and be affected by it. So we all have 80 to 90 billion neurons, and most of them are actually produced by mid-pregnancy. Neuronal migration, that is neurons getting to the right place in the brain to have the circuits we all need to function. That migration peaks at mid-pregnancy. There are 40,000 new synapses formed every second in the third trimester. That's the peak period. And what I can say is that most of us in this room would give anything to have that many synapses forming every second. A synapse is what connects a neuron to another neuron so that our brains can really function and communicate, as many of you know. So to really put this into perspective, you can see the timeline here on the bottom and all of this brain development that's going on in utero relative to later in life. That really indicates that the fetus, there is brain activity going on, can register the mother's experience because the mother's experience is his or her environment and therefore be affected by it. So the fetus registering the mother's experience. Newborns suck more to the scent, which is an indication they prefer their mother's breast milk to that of other scents of breast milk, based on the continuity between the olfactory smell cues in the amniotic fluid environment and those in her breast milk. So it's familiar. Newborns suck more. They prefer a tape recording of their mother's voice versus a female stranger's reading Dr. Seuss, because after 9 months in utero, their mother's voice is very familiar. Pregnant women who exercise have fetuses with healthier heart rate patterns. And also, even maternal women's emotional experiences are registered by the fetus and affect them. So an example of this, in one study from 1967, a pregnant woman was told that she was breathing a gas which contained only half the amount of oxygen necessary to support fetal life, but that her normal body mechanisms would probably compensate for this altered condition. Whoa, that's pretty scary. So what happened when this woman is given a fright, a stimuli, a psychic stimulation, as these experimenters, these researchers are going to call it? It's a fright response for her. She's, of course, very activated by this. What I'm showing you here is a tracing of her heart rate pattern on the upper panel, and on the lower panel is the fetus's. And to that psychic stimulation of being told maybe the air isn't right for her baby's life to be supported, her heart rate goes up, just as it does for all of us when we have a fear experience. And what happens to the fetus's heart rate, coincident with the mother's heart rate? It goes up as well. So the fetus is registering the mother's acute emotional experience. So you've already heard a foreshadowing of this. It's sometimes called DOHAD research, and here I'm calling it FOAD research, developmental or fetal origins of adult health and disease. So not just acutely, as I just showed you in that 1967 study, but over pregnancy, women's experiences affect the fetus and shape their future brain behavior development. Now this topic made the cover of Time magazine several years ago. And this is a really felicitous phrase that captures this concept of FOAD research. Think about this, how the first 9 months shape the rest of your life. So what happens if there's stress in a woman's life? What happens if she's living in adverse conditions, chronic poverty? What happens if she's experiencing toxic stress, that ongoing chronic stress, anxiety or depression? What happens if she has a psychiatric illness? That's an influence on the future child's neurobehavioral development as a fetus, prenatally. And here's some papers I want to describe to you that really support this idea. This is a paper that came out years ago on a study called the Allspack study. It was a prospective study of 4,000 pregnant women and their children. And in this study, the kids are 6 and 7 years old. And what you're seeing here in this table, which reports on the data from the study, is that when women had higher anxiety, antenatal anxiety during pregnancy, where I've circled in red, their children at 6 and 7 years old were more at risk for having a psychiatric or psychological problem, attention deficit disorder, or a behavioral problem, years later. And in another study with children about the same age, also looking at anxiety, but a smaller study, this sample is only 25 subjects, because they were using brain imaging. And what they found here is that when women had higher anxiety during mid-gestation, when their kids were 6 and 9 years old, between those ages, they had less gray matter density. That means they had fewer neuronal density in their brains. And this finding was in very important areas: the prefrontal cortex, the premotor cortex, and the medial temporal lobe, all areas very much involved with how we regulate emotions and our behavior. And finally, in a study with younger children, but very important in terms of how we manage our stress regulation, these researchers were able to get at the level of cortisol, that stress hormone, in the amniotic fluid of the mother while she's pregnant, and then follow these kids when they were 17 months old and give them a stressor in the lab. So children who had low levels of amniotic fluid, of cortisol in the amniotic fluid, they had, if you will, the correct response. Their cortisol went up to stress, which is what it's supposed to do. And you can see that in the non-hatched line. But the children exposed to high levels of cortisol in the amniotic fluid, they had a flat line or a decrease. Their HPA axis, the hypothalamic pituitary adrenal axis, is not regulated correctly. And that's just from what they were exposed to in utero. So pregnant women, again, you're not seeing it all. I'm sorry about that. Pregnant women with toxic stress have children who are more likely to go on and have neurodevelopmental problems and alterations in their HPA axis. In my group, we've been looking at how fetal studies can add to our understanding of FOAD. Just as you saw in this 67 study, it's what we're working on. And obviously, when we look at the fetus, it's independent of post-birth experiences. So we can see the maternal toxic stress affecting the child at the time it is believed to occur during the fetal period. So this is one of our participants. She's in our lab, and we're getting lots of biological information from her, as well as getting information on the fetal heart rate, just the way you saw in that 67 study. But for us, because we have institutional review boards that don't allow us to do unethical things, we don't tell her that she's possibly putting her baby's life at risk. Instead, the psychic stimulation is a cognitive task. It's a Stroop color word matching task. Many of you may know that from psych classes. It's really a cognitive challenge task. And I'm going to show you a complicated graph now of what the fetus does when the mother is having this psychic stimulation. And I'll walk you through it. What we're seeing is that some fetuses, particularly those exposed to toxic stress, had a fetal heart rate reactivity when their mothers were doing the stress tasks, and other fetuses didn't. Particularly, you can see in the green, those are fetuses of mothers who we're calling control. They did not have toxic stress. They did not have a diagnosed psychiatric illness in their lives. But in the purple and the red, in particular, those are women with comorbid anxiety and depression disorders, or they have a depression disorder. And for those fetuses with that toxic stress exposure, they are more reactive during this psychic stimulation. And then we went on and looked at, does this fetal heart rate reactivity, being more reactive as a fetus, tell us anything about what the brain is doing using MRI when the baby is born? And what I'm going to be telling you about here is functional connectivity in sleeping babies, so resting state functional connectivity. And there's the title of the paper, and as you can see, this is very much a group effort, as we were just talking about with Team Science, and you see the names of my key co-authors here. Looking at the amygdala and the prefrontal cortex, the amygdala, of course, being very involved in our fear responses and regulating emotion in general, and the prefrontal cortex helping us at times to dampen it down and say, no, don't be afraid, it's really just a shadow, it's not someone coming after you. So the prefrontal cortex, the amygdala, in an adult work in an inverse pattern to help us manage the kind of stimuli that comes our way. What we saw in these newborns who'd had toxic stress exposure from their mothers during pregnancy in the form of untreated depression was that they too had this inverse pattern. They showed the resting state connectivity that was the pattern for adults, this inverse connectivity between the prefrontal cortex and the amygdala. And very interestingly, we saw strong associations. So the fetuses that had the strongest heart rate reactivity when their mothers were undergoing psychic stimulation in our lab, you can see they had the strongest inverse connectivity in their brains at birth. And we also saw that the fetal heart rate reactivity helped us predict, it told us what kind of babies they were going to be at 4 months old in terms of their temperament and how they responded to novelty. So this is work carried out by my close colleague, Liz Werner. And we saw that the greater the fetal heart rate reactivity during the psychic stimulation of the mothers in our lab, the more reactive the babies were at 4 months to stimuli. And I'm going to show you 2 tapes, and I want you to tell me which baby is the more reactive baby. Oh, we're not going to get our tapes to play? Oh, there it is. This is a quiet one. Cute though, right? All right, you got your vote, but wait, let's see the contrast, or if there is a contrast. What do you think of this baby? This one happens to be just a louder tape. All right, what's your vote? Who's the more reactive, 1 or 2? All right, okay. Everyone gets an A. So what do we make of all this? I'm going to put together what I've already described, our fetal and infant studies. Prenatal maternal toxic stress is associated with what we're thinking of as accelerated development. In the fetus, we see they're more reactive to maternal laboratory psychic stimulation. They have greater heart rate reactivity. They're registering changes in the environment when other fetuses are not. And in the infant, we see greater inverse functional connectivity between the amygdala and the prefrontal cortex, that adult pattern. And in the babies we just looked at 4 months old, they're more behaviorally reactive to novelty. They're more aware of their environment, even maybe more vigilant. So this idea that maternal prenatal toxic stress is affecting the fetus has now been thought about from an evolutionary perspective. Why might this be? From an evolutionary perspective, is there some purpose being served for our species? And the thinking is that the maternal toxic stress exposure is forecasting an adverse environment for that fetus, that future child. So the fetus is programmed to have accelerated maturation. It's going to be adaptive for this fetus. Be prepared for the stressful postnatal environment to come. I'm feeling toxic stress right now. I'm going to forecast and communicate to you in utero to develop, adapt in ways so that you're prepared for the stress that you're going to encounter once you're born. So what are the consequences for the child? Well, they might develop a style, as we've already seen, where they have a heightened awareness, a reactivity, a vigilance. But there's the possibility that at times it's really a bad match with the environment. It's a mismatch with their postnatal environment. So if think of that second baby, think of a more reactive fetal heart rate. If that's your way of being in the world, you've been programmed for danger through toxic stress communication from your mother, and you're a 6-year-old boy, and you come into a classroom in the 21st century, and like me right now, I'm supposed to ignore that, right? You are supposed to ignore everything else in the classroom and stay focused. But you weren't programmed that way. You were programmed to be adaptive and ready for all that comes your way in the toxic stress environment. So it's a mismatch. And what do you look like you have? Attention deficit disorder. You can't pay attention. You can't take out the stimuli. You were ready to be alert to all that stimuli because that's going to save you in your dangerous environment. So this idea that maternal prenatal toxic stress exposures are communicated to the future offspring to help the species has been very much supported in animal research. So you see, of course, a very adorable squirrel here, and this is a really interesting paper, and I'm picking one of thousands. With these pregnant squirrels in this study, they used auditory cues so that the pregnant squirrel, and particularly the communication to the offspring, was that you're in an environment that's really populated, and getting food is going to be hard. You're going to have to really fight for it. So in utero, those baby squirrels actually had accelerated physical growth, the better to prepare them for fighting for the food. But in fact, their environment was just fine. So they were really able to use experimental control to show that when the mother is thinking, just has the idea, I can't get enough food, she's secreting a hormone, and through that hormonal exposure to the baby in utero is shaping their development to be ready to fight for food. So that really does bring us to the question of transmission. How might it be, and I've done a bit of foreshadowing, but how might it be that a maternal experience is transmitted to the fetus? And this is really a map. We've got this maternal toxic stress. What's the transmitting signal that's altering the in utero environment, affecting fetal development, and putting that child at risk for psychopathology? Well, stress, as many of you know, and I've already alluded to it, is this toxic stress can be associated with differences in the stress hormone cortisol, and that is a glucocorticoid hormone. I'm saying these words because I'm going to show you a paper with a lot of these terms in it and then describe the research. Again, in team science, these are two of my colleagues. We worked very hard on this paper together. And here we're going to be looking at epigenetics, and I'll define that, and what's going on in the placenta as a pathway by which maternal stress is communicated to the fetus. So first of all, epigenetics, as most of you know, is a change in gene activity, the expression, without affecting the basic structure of the DNA of the gene. And the epigenetic mechanism I'm going to be talking about is DNA methylation, which most often is associated with turning off the expression of the gene, so making the gene so it doesn't function. So what we found, and I guess you can read that, that's good, is that maternal toxic stress during this first period of gene-environment interaction, this in utero period, turns off a placenta gene that deactivates cortisol as it crosses the placenta. That's going to influence fetal brain development by altering the amount of cortisol in the in utero environment, and that affecting fetal development and the child's future health. And I'm now going to show you some raw data from this study. What you're seeing here is the name of the gene, HSD11 beta 2. And on the y-axis is PSS. It's a measure of stress in the woman's life. And on the far right, you see that the higher the PSS scores are, the more this gene is getting methylated, so it's turned off. So then the active cortisol is crossing over to the fetus and affecting brain development in the context of greater stress, turning off that protective gene. All right, so we have a relationship during pregnancy between the mother and child. This is a prime opportunity for intervention for both her and the baby. As we saw, the fetus registers and is shaped by maternal experiences via communication or biological transmission, likely all of them. We saw this in maternal scent recognition, maternal voice, and stress affecting gene expression in the placenta and having implications for baby brain development. Oh, but you're really missing the last part here. The baby is in the mother's mind and heart long before birth. She's already envisioning her relationship with her child. So in a project with my close colleague Liz Warner, we've been taking a dyadic approach, a mother-baby approach to reduce maternal prenatal toxic stress, support the mother-infant relationship, and thereby prevent postpartum depression. So this is an intervention based on the conceptualization of postpartum depression as a potential disorder of the dyad, of both mother and baby, and one that can be approached through preventative psychological and behavioral changes in the mother that affect her and the child even before birth. So taking a minute to look at prenatal toxic stress and postpartum depression, toxic stress, as we've discussed, affects the fetus. Toxic stress is a risk factor for postpartum depression in the women. And about 10 to 15% of women experience postpartum depression. And as many of you know, postpartum depression also negatively influences children's social emotional development. And really importantly, women who are living in poverty are at twice the risk of postpartum depression. Their rates are much higher than women who are not facing such adversity. So our intervention is called PrEP, Practical Resources for Effective Postpartum Parenting. And it's a very brief intervention. It's 4 sessions. It starts in pregnancy, and it's time to have this intervention, this coaching, happen while at the same time that the woman goes for her OB appointments and also her well baby doctor visits. And what we're doing here in thinking about potentially depression as a disorder of the dyad, we're thinking about how much the mother and baby affect each other. And so if you start with one on this loop of bidirectional experiences between the mother and baby, part of PrEP is coaching the woman to have many behavioral techniques to help her baby have as best regulation as possible, behavioral regulation. And that is through giving her different tools to help her baby sleep more and fuss and cry less. And if you follow the circle around in one direction, as we're optimizing the baby's sleep and fuss behavior, she's going to feel a more effective parent. She's going to feel competent, positive about her baby. That's only going to add to her sensitivity with her baby. And that in turn is going to help her baby be even better regulated. And if you go down the other side to the right in our bidirectional circle, as the baby's better regulated, the baby is sleeping more, the mother is sleeping better. The better she sleeps, the less distress she has, the more likely it is that she's protected against postpartum depression because she's better regulated. We're also intervening in the mother-baby influences on each other by direct care of the mother through psychoeducation and mindfulness and self-reflection skills. So we use a brochure when we do our intervention, and I'm just giving you a couple of pictures of what this brochure is like. And I'll show you some evidence of how PrEP has already been found to be effective. So in women with severe toxic stress in pregnancy, they're at high risk, as I've already indicated, for postpartum depression. In fact, about 40% of them go on to have postpartum depression. And in fact, in our enhanced treatment as usual, in that one arm there, about 40% of them, those who did not get PrEP, went on to have postpartum depression. And we cut the rates in half for those who received PrEP. And then as we predicted, with PrEP, the women rated their babies as fussing and crying less and sleeping more. That sleep is not on the graph. So some concluding thoughts. We can ask the fetus questions and see the effects of maternal toxic stress. Beyond shared genes of risk interacting with the postnatal environment, there is a possible third pathway by which maternal toxic stress affects children's development. And that's through pregnant women's experiences of toxic stress affecting, and you're not seeing this because the sentence is cut off, but really importantly, affecting that baby before they're born. Depression, stress, anxiety are treatable when there's access to decent behavioral health care. So we can help pregnant and postpartum women, and by doing so, help the next generation, help those future children. So this is a picture of my lab. It's the Perinatal Pathways Lab, and as I've already indicated, research always happens with a team effort, and these are some of the folks working with me, and I want to thank our funders as well. And end on one of my favorite cartoons. I like to think that each generation will need a little less therapy than the generation before. So thank you very much. Great. Am I able to, I can just go ahead and take questions? Okay, I would welcome any questions or comments. Yes, please. Yes, go ahead. Hi. Wow, that's a great question. We've actually extended it, I should say. So we now, but I didn't tell you that. So we have 2 sessions in pregnancy. We knew we wanted to start in pregnancy at that time when the woman psychologically is already really preparing, and also because we want to interrupt that toxic stress effect on the fetus, right, while it's happening. So we wanted to start in pregnancy. We knew if we went way too far back, it might be a bit odd because we're giving them carriers to carry the baby. We actually practice with the doll. So we wanted them to be pregnant enough that it's really pretty secure that the pregnancy will hold, and that it didn't feel too far off that the baby was coming. But we wanted it to be a brief intervention. Obviously, some women are going to need more, but we really wanted to show, and we are continuing to do this work, that a little bit goes a long way. And you can get a mother and baby on a good path with fairly few sessions. Okay, welcome. Who else? That was a first courageous, yes. So the Stroop task, does anybody, I bet someone here knows the Stroop task. Anyone want to answer? You remember this? It's a color word matching task. So it's a color word matching task. So you're shown in our version of it a color word, say the word red, but you're shown in blue letters. And your task, is it coming back to some people? So your task is to actually answer the color of the writing of the letters. But because we read so automatically, it's very confusing. And in our hands, we're not as cruel as they were in 1967, but in our hands, we have people in white coats telling them you're going too slowly. But they don't know that if they actually do it well, the computer speeds up and they have to do it faster. So it's pretty stressful. It's only 5 minutes. And it's not life-threatening, but it's stressful. Does that answer your question? Yeah. Yeah. Right. That's a great question. Just to be sure everyone could hear, the question was looking at toxic stress effects when the pregnancy is a twin pregnancy. So we don't do it just because we can't. I wish, if we could somehow label twin A, twin B each time, we could know who's doing what over time, but of course that's hard to do. There are some studies, looking at twin pregnancies, and of course in many ways just physically they're riskier pregnancies at times. What's interesting about twins is that even when they're sharing all the same genes, they actually have different developmental courses, as you seem to indicate. And so that just underscores that the in utero environment, one is getting a little more oxygen, one's getting a little more nutrient. There's slight differences that are contributing to the developmental paths. But I'm not doing that yet. We're not able to keep them straight, to follow them. Yep. Yes.
Still fascinating.
Thank you.
Lots of questions.
Yeah.
My one question now is, you did stress, but what happens if a mother, an expecting mother, has overwhelming happiness?
Yeah, great question. What a wonderful question. Again, I just want to be sure everyone's hearing them. Instead of stress, what about if you're exposing the woman to happy, positive experiences? We are right now doing a relaxation paradigm, and we're looking at when the woman relaxes, what does the fetus do? And we're also building on the scent recognition. We are pairing the relaxation with what we would call now an aromatherapy. And because the scent is in a lotion, that's going to get into the amniotic fluid, and we're assuming that the fetus, we're hypothesizing the fetus, when he or she becomes a baby, is going to have the exposure of the mother relaxing, opposite of the psychic stimulation, paired with the scent, so that we're going to call them back and do EEG on the baby while the mother's relaxing with the scent again, and see if we get more of a relaxation in both of them than when we don't have the scent. So we're trying to build on that. So it's a great question. We can induce different experiences. Yes. Yes, yes. And we should be looking at these positive, protective experiences and showing how they affect the fetus and future child as well. Absolutely. Yeah. Go ahead.
Okay, I have two questions.
Great.
One, I'm not sure if I missed it, but how exactly do you define and, I guess, measure the toxic stress?
Yep.
And then the second one is, if the toxic stress somewhat is when you're talking about, like, bringing it back to the past, if it somewhat is bringing out this fight or flight response in these children, could that sometimes be a good thing depending on the environment that you're in? So
Right.
if you're born maybe not in the United States, but somewhere else where you have to be raised in this type of,
Yeah.
you just fight for yourself environment, maybe this is a good
Right,
thing.
right. Wow, those are both great questions, and I'm assuming everyone could hear because you're up high and your voice is coming down. And I have to see if I can remember both because they were really good. The first one is, in essence, you're really asking a question like a researcher, which is how, I want to use the term operationalize. I'm telling you we've got a concept of toxic stress, and you're asking me, how did I measure it? How did I operationalize that? In our studies to date, the ones I showed you, they're diagnosed psychiatric illness. And they're untreated. So we're doing a psychiatric interview, and we're getting all that information. There's certainly other ways to do it, but that's what we're doing here. And then absolutely being programmed, adapting in utero to a dangerous environment. I probably should use the term microenvironment because it might not be very adaptive in certain classrooms, for example. But when you're out on the playground, if you're living in an adverse environment where there is a lot of danger or bullying, having your wits about you could be very adaptive. So that's absolutely true. And that's really why the idea of evolutionary theory has been applied. Why would it be? The fetus is very protected. But why would it be that these experiences of stress are getting communicated to the fetus, or the experience of maybe there's not enough food? That probably serves a purpose to help us get ready. So I think in certain microenvironments or even macro environments, depending if you grow up in a war zone, could serve you. Yeah. Great, yep.
Is there also a correlation between the toxic stress the mom experiences with the fetal movement? So it's like they may
Right.
go up or effectively
Right.
under more?
Great question. So the question is, can we see a parallel between a toxic stress exposure and more reactive fetuses in their heart rate? Can we see a parallel outcome in movement? And the answer is basically yes. Movement is much harder to measure in the fetus accurately. So we do look at movement in the fetus, and it tends to track with the heart rate, just the way when we're activated, when we're running, that's when our heart rate is up, or when we're physically doing things. And you can see also pulling away, and you saw the more reactive temperament. It's just literally a less reliable variable for us. We can't count on it as well. But I don't yet have, I think the other part of your question is, would that predict to more physical behavioral outcomes? And I do know of one study by Janet Pietro who's looked at, she hasn't looked at movement. Again, it's a hard measure in the, no, I beg your pardon. She did look at movement, and she saw that it was associated with being less fearful at the age of 2. So a little bit opposite of what I'm telling you. But that was more, that was being, pretty active movement-wise. So you can wonder about different paths leading to different futures. Good question. Yeah. Hi.
Thank you. Great presentation.
Yeah.
I have 2 questions. One was more specific about the graph you showed with 43% versus 23%. What was the sample size of your study? I was more curious from a statistics standpoint.
Boy, the 43 versus 23.
Right, you said like they had a better outcome, 43% had a better outcome.
Oh, prep. Okay, wow. Okay, run it. I was like, wow, where was that figure? Now ask me the question again. Excellent question. So these are great questions of budding researchers or people who've had a good bit of experience. Great question. That was a relatively small sample size of 54. It was randomly assigned, so it was an RCT. And we're currently doing a much larger study with 215 women who will be randomly assigned.
And then just a follow-up to that
Yeah.
question was including those women that you looked at, like you said in the question before, they had a diagnosis of a mental illness.
Yeah.
Were any of the mothers by any chance happen to have a diagnosis of substance abuse disorder?
No.
What's your opinion on looking at this study with that population?
Right. So that's a great question about substance abuse. Again, I'm assuming you all hear it because it's coming from high up in the elevation. It's hard to study women who have substance abuse disorders because they're very reluctant to be in a research study, because even if I tell them I'm not going to report them, et cetera, there's a lot of fear. Also, not uniformly at all, but a lot of times during pregnancy, women actually do lessen their substance use. There's a huge motivation for the health of their baby, so they're actually hard to recruit. I have a postdoc working with me right now, and she's really got excellent data, for example, on alcohol exposure in utero, and I think this is going to be quite surprising to people. But when you get rid of the other adverse psychosocial factors that are associated with drinking alcohol, for example, in pregnancy, or maybe taking substances, we were talking about opioids at dinner. Opioids is probably is a different animal, let's say, but you don't find a lot of effect of alcohol unless it's just a huge binge drinking. And so you have to wonder, is the alcohol a marker for other factors in the environment that are really affecting fetal development, or are they working synergistically? So something about the chemical makeup of alcohol enhances the exposures that can be very negative that go with the alcohol situation. But I haven't looked at it personally because we don't end up recruiting those women. Our women don't even smoke. Yeah, great question. I saw a bunch of hands, and yeah, go ahead. Yep, go ahead. Yep. Right. Right. So that's a great question. What about maternal age, and particularly with teenagers? So for, let's see, 7 years we were doing a study, and we're still working on the data with pregnant adolescents. And part of our argument was that this was going to be a group that, unfortunately, but the reality was, would have enhanced stress. And so that would improve our opportunities to detect effects. But one thing we're doing right now is actually putting our data sets together with the adults and the adolescents. And there can often be the opposite of what you're asking. So you're asking about being super young, and we are seeing some effects. But the other question that comes up a lot is being an older mother not a good thing. And what we're actually finding is it's really, for reasons we still don't understand, seems to be associated with more optimal development. And that's very counterintuitive, because a woman who's over 35 is just automatically deemed at risk because she's an older mother. So age is really interesting, but you have to wonder, again, what is it a marker variable for? She's lived a longer life, the choices of when she had a baby, what are we really getting at with that variable? Because it's probably, when I say a marker, it's standing in for a lot of other factors. Great question. Yep. Yeah. Yeah, I mean, I'm pretty cautious, and I would say actually I'm very cautious. I don't have concern. I mean, I think there's a resource problem around MRIs, so a little bit of a tangent, but I have a project right now that goes on in Puerto Rico. So these, with newborns getting imaging, and they're non-sedated, and we swaddle them, and they sleep very happily in the scanner. There's a resource problem, which is if I ever detect something questionable on a newborn's scan, I can very easily in New York get that baby a follow-up scan. There's a resource problem in Puerto Rico where that doesn't happen as easily. So I find that a problem. The issues around MRI, I don't think there's anything to be concerned about. And as I say, I'm pretty cautious. And in fact, I'm part of a project run by Mariah Thomason, who's at Wayne State, and she's actually doing imaging of the fetus. And so people are pretty convinced, and I'm not a physicist, but when I listen to the physicists talk, it's very convincing that this is really safe methodology. Yep.
Do you think that the physiological relationship between the mother and the fetus is bi-directional?
What a great question. I think a lot of you read my mind. You're thinking about things I'm thinking about. So the question was, do I think it's bidirectional? And the answer is absolutely. And there's just beginning to be research on this. So if you're carrying a female, we have several pregnant women in the audience. I don't know what you're carrying. If you're carrying a female, you tend to have higher cortisol, higher stress hormone. Janet DiPietro, again, I've quoted her before, she's very involved in this kind of research. She showed that using galvanic skin response, which is a register of the sympathetic activation, that when women had fetuses that moved more, this goes back to your movement question, they were more sympathetically activated. Now, the intervening variable here that was of interest is they actually were more anxious. And there have been associations, and I should have said this before, between more anxiety and more fetal movement. But what she said is, maybe it's not that they're actually anxious, but the women are feeling this sympathetic arousal and saying, and they have to label it, oh, I'm anxious. So there's the fetus affecting the mother through sympathetic arousal, and then the mother having a label for her subjective experience. So there's much more to come in this. I think you're going to be reading a lot more about this bidirectional. Yeah, I'm really interested in it. Yes? Yeah. Right. That's a great question. And these are all such great questions. I have to find another word for great. It's a very good question. And it was about medication, psychotropic medication being used to treat, to help the woman with her anxiety or depression. In all the data I showed you, none of the women were on medication. However, and I don't have these slides on me, I'm really at the very end of working on a data set of 161 pregnant women. All of them had a diagnosis of major depression. 70% of them were on medication, and very well treated. So actually the depression was really low in these women on average. They were getting a lot of treatment, a lot of help, both psychotherapy and medication. And I have to be honest and say we really went into this saying we're going to find effects of medication, because the epidemiologic literature, meaning the large population studies or certain cohort studies, have sometimes found associations between, for example, SSRIs, and autism risk, or depression in the offspring. And then the challenge to these data sets has always been, well, maybe it's the underlying depression itself, and that the women who are having such a difficult time that they are on medication, it's the illness, it's the psychopathology that's in body and mind that is actually affecting those children. So we thought we'd go in and look at fetuses, and if we could see it happening in the fetus, we'd be able to help this debate. And I have to say that, and we were just on the phone the other day about this, a big project with people from Emory University, Zach Stowe, it was his project, he was the lead. We have worked on this data so hard, and we don't see anything in the fetuses. And then we looked at the babies at 12 months with a Bailey, a cognitive and motor task on the Bailey, and we don't see anything. Slight is an increase in movement associated with SRI, and the thinking is and then they sometimes show that right after birth. They're a little more, they're less regulated. And the thinking is that it's still the medication on board. My way of putting the literatures together that disagree with each other is that SRIs, as you all know, are they're part of acting on our serotonin system. And serotonin during brain development functions like BDNF. It's a neurotrophic factor. It helps neurons grow. And so other people, Tim Oberlander, Gurr at UPenn, have the idea that if you're getting exposed to more serotonin in your brain in utero, it's maybe not a bad thing per se, as much as it's increasing the plasticity of your brain to be open to the environment. And so depending on the environment you encounter prenatally and postnatally, it could have better or worse effects. So are you an orchid brain baby? You're very sensitive to whatever you get. So if it's good, you're beautiful, you're really beautiful and thriving, and if it's bad, you really wither. Or are you a dandelion? You know, you can take anything and you're about the same. And the idea is that being exposed to an SRI in utero may make you more of an orchid. And so then it really matters what you're exposed to. So that's some of the current thinking of how to put these literatures together, and I think our fetal data, which we're about to submit for publication, supports that. Stripes.
Are you guys looking at preterm and preterm, like NICU babies? Do you think that with heightened, fight or flight response would be much better for, the clinical research?
Yeah, that's a really interesting question about preterm babies, and part of the thinking is that they get out of in utero because in utero in some ways is adverse, right? And they want to get out. I'm not myself looking at preterm. There are many programs at Columbia that are, and it is another way to study brain development because and brain behavior development, I should say, although you're out of utero and there's a big difference between the postnatal environment and then utero. But so I'm not myself, but there are a lot of studies. Great. And yes, go ahead, sir. Yeah. So. Yeah, so the question is, have we followed any of these children? We're just submitting a grant following up on our pregnant adolescents, where we saw those fetuses starting in the second trimester, and we're followed them up to 2 years old. And now we're just about to submit a grant so that we can start studying them again around 5 and 7 years old. So we would really like to do that. The life of most grants is 5 years, and then you have to get more funding. So this is a challenge always in research, but we would like to do so. So instead what we can say is we can insert our fetal story, and other people have similar early development stories, with the other papers I showed you before to try to make a logical argument, but it's hard to study the same kids for so long. But this ECHO project, and for all you folks in training right now, I really suggest that you go online, look up NIH ECHO, and this is the project you heard about that's going on for 7 years. It's going to be an open data set. That is the obligation from NIH right now, and it's going to include placentas, brain imaging, also, blood, all sorts of other biospecimens from the mothers and the children, and they're going to be followed and followed. The idea is to really make this a birth cohort in the U.S. so we can really look at the same kids over time. Yeah. Yeah. Yeah, you mean, that's interesting. So the differences in the labor and birth experience and how that, people have done that, and there are just so many different factors, and they've not seen so much, and also medical care can vary in terms of how a situation is handled. I'm going to flip your question a little bit and say, I have a colleague who gives psychic stimulation to pregnant women in her lab by showing them a film of labor and delivery. And so that's very stressful. Yeah. You let me know. Can I take a few more? 2 more. Okay. I think you had one, so I want to, and you had, was there someone? Go ahead. Yep. Yeah, let's see if I remember. I've been challenged in my memory. Go ahead. Oh, you're looking online. Very good. Okay. Yep. Yeah, absolutely. Right. So the question is, when we look at demographics, have we considered doing a better job of looking at socioeconomic factors? And I'm personally very interested in what poverty does and how it gets under the skin and affects us and the next generation. And I'm thinking right now in the back of my mind about a finding and how to explain it. So we haven't done that as well as we could, and it does interest me. Bear with me for a minute on this. There's a way to use epigenetics to look at what's called the epigenetic clock. So on a cellular level, what is the age of the cell? And so you can look at people in poverty and pick some peripheral cells, and they're actually older, much older than their chronological age, which speaks to how adversity has gotten under the skin. So we've been looking at this recently in the placenta, and using this epigenetic clock calculation, and I was really intrigued to see that one of the variables that really came up was income. And actually not the lowest, because in pregnancy, it's one of the few places where our federal and state governments still give you some resources, so you're automatically on Medicaid. And I don't mean to say that's enough, but in this sample, it was the notch above who are still really struggling, but their placentas really showed the effect, their strongest association. So I'm really curious to pursue that. So great. Another good research question. So I get one more? One more. Okay, yes. I saw that, so anxious. Right.
I was thinking that, do you think that the kinds of things that you're seeing are more, is there any way of knowing whether those things are more common now than they were in the previous generation?
Yeah. That's even the question, right, with autism. You know, is autism rates actually going up? Are we better at ascertainment and labeling? So there was a piece in the New York Times today, which I have not yet read, about why are the rates of significant anxiety in people your age and a little bit younger supposedly a lot higher. And it's so hard to see. Are they truly higher, or are we just more aware of it? But I do wonder if they aren't really higher. And for a lot of the factors going on in the world, possibly that there's been a lot of anxiety for decades now, and so part of it is getting communicated in utero. If I can make a bad joke, my mother was able to drink during pregnancy, so you know, having a martini in the afternoon could really take away. It may have affected me in other ways, but that's so it's a very interesting question. But you know, obviously, and one thing I should emphasize is that these prenatal factors are there. They're certainly not the whole story, because we go on being exposed and interacting with our environment. But it may be a piece of that, and I'm curious to read it. Yeah, great. Okay. Thank you. Thank you. Great. Thanks. Oh, I'm so glad. Oh, you've just been such a great. Oh, such a pleasure.